What can microbotanical remains tell us about ancient human diets? Imagine tiny plant particles so well preserved that they can tell us exactly what our ancestors ate thousands of years ago. Microbotanical remains include starch grains, phytoliths, and pollen, and they are like microscopic clues hidden in archaeological sites. These tiny remains are often found trapped in soils, on artifacts, or in dental calculus, which is calcified plaque on ancient teeth. Dental calculus is especially useful because it captures food particles during a person's lifetime and keeps them safe for thousands of years. When scientists analyze these remains in the lab, they can identify specific plants based on the shape and structure of starch grains and phytoliths. For example, certain starch grains have unique shapes that can tell us if ancient people ate roots, seeds, or nuts like acorns. Phytoliths are silica structures from plants that also have distinctive shapes, helping archaeologists distinguish between grasses, reeds, or other plants. This detailed identification helps us understand what kinds of plants were part of their diet. Microbotanical remains also reveal how diverse their diets were. They can show whether early humans relied mainly on wild plants or if they started using cultivated crops. This helps us see how their food choices changed over time and across different environments. Analyzing these remains can also tell us about food processing techniques. For example, changes in starch grain shapes can indicate cooking, grinding, or fermentation. If starch grains look broken or altered, it suggests the food was processed in some way before eating. Comparing microbotanical evidence from different sites and periods helps us track shifts in diet, like the adoption of new plants or farming practices. It also shows regional differences in what people ate, based on local plant availability. Microbotanical data complements other archaeological evidence like animal bones or larger plant remains. When combined with chemical tests, it gives a clearer picture of ancient diets and how humans interacted with their environment. Because plants respond to climate and landscape changes, microbotanical remains can also tell us about past climates and how humans adapted their diets to new conditions. Collecting these tiny remains requires careful sampling and lab work, often involving washing, filtering, and microscopy. Analyzing dental calculus is especially helpful because it provides direct evidence from individual people without damaging their bones. Overall, microbotanical remains are a powerful tool for uncovering what ancient humans ate, how they prepared their food, and how their diets evolved over time. They help us piece together the story of human life through the tiny plant particles that have survived for thousands of years.